There you go.
Okay, and I'm loud and clear. Yes. Okay, welcome all to today's webinar, where I will talk about the Bridge Sweden project and our new cell line development facility. Can I maybe remind everybody to mute? I don't know if you have that power, Henrik. Okay. So then the Bridge Sweden project is a Vinnova financed project spanning over 3 years that aim to help projects moving from the discovery phase into the developmental phase of drug discovery. And we are many different partners involved in this project. We have the Drug Discovery and Development Platform at SciLifeLab, Stem Cell Center and the GMP facility at Lund University, all acting then in the discovery and optimization phase. We also have Testa Center in Uppsala that are working with the scale-up phase, and we have the different innovation offices, Stuns and Umeå Biotech Incubator, that are working with selected projects throughout the project line. And we also then have here a few projects that are selected to get the support in the Bridge project, and two of these, the Tuitran and Anna Dimberg, are antibody-based projects. They have very interesting antibody candidates that then will get the full sort of cell line development support within Bridge Sweden. And I am then working at the Drug Discovery and Development Platform. And just to give you some background on what that is, it's Sweden's national bridge from biological discovery to therapeutic reality. So we are spread out over all of the different SciLife facilities, and the idea is that academic ideas come into the Drug Discovery and Development Platform. They go through a DDD consultation. A few of the ideas are selected for a full DDD program where they work with project leaders, the DDD platform, to fully develop their drug candidates. And we work with different modalities, but for the sake of this webinar, I'm mainly going to focus on then the antibody therapeutics, and that is also the unit where we have now the cell line development capabilities. And we have seen quite a few success stories going through the DDD platform with Lipum and Acrium Therapeutics, to name a few. And if you think that you have a that could need industry level of support in developing your drug, please contact us or follow us on LinkedIn to see when we have open calls. So that was the Bridge project and the DDD platform, but today's webinar is revolving around cell line development. And since we have a quite broad audience, I think I will go in quite basic to just outline what cell line development is, and also on what to think about when thinking a cell development provider, and then also say what we now have to offer within the Bridge Sweden project. So then, looking at the classic drug discovery pipeline, you start with your target identification all the way up to your lead optimization in the drug discovery phase. And then you have the second part, which is the developmental phase, when you have your one candidate that you want to take into the clinic. But going from the drug discovery phase into the developmental phase, you have one sort of crucial thing that needs to be delivered, which is the cell line development. And now again, I'm talking about recombinant protein drug discovery pipelines. And this cell line development then is a quite time-consuming process where the aim is to generate one host cell that is producing your drug substance for the rest of the lifespan of the drug project. So since we are then using living factories for the production of our drug substance, it is very important that you follow a outlined a precision manner to get a very good production host. And I thought I'd highlight a few parts in this process. So then, of course, you start with your gene of interest that includes your protein of interest that you are transfecting into your selected production host cell. When it comes to recombinant proteins and antibody production, this host cell is almost always a mammalian cell line, and most often it is a CHO cell line. After the transfection, you will go through the selection process. We have one cell that is selected that show good production capabilities that you later on then expand and then move into GMP banking. And this is important to say that the whole cell line development process is a non-GMP setting. It's first when you move into the cell banking and manufacturing that you go into a GMP setting. Now then, to highlight two of these parts in order for you to understand a bit more on the cell line development phase and what different aspects you can take into account, I thought I'm going to start with the transfection part. So there are many different ways to integrate your gene of interest into your host cell. One of the oldest and most commonly used one is integration. So there you have your gene of interest and your selection marker that via transfection is included within the genome at random locations within the genome. And this can be either as multiple expression cassettes that are put together, giving a quite high expression, or at multiple locations, also giving high expression. But it can also be that they are at a quite low expressing regions within the genome. And this means that your selection process, where you go through all of the different clones in your stable pool, is quite tedious. You will have a very diverse cell pool, some of them expressing very low, some of them expressing quite high. So the selection process with random integration is quite tedious. Another system then is the semi-targeted integration system. This system utilizes transposases to integrate your gene of interest into specific motifs within the genome. Now, these motifs are known for being in quite well expressing regions. So usually you will get a quite good cell pool with a more homogeneous characteristics of the cells and expressing higher titers. So this also limits the amount of selection needed to be done, but it also generates a more homogeneous pool if you would utilize that for other parts in your developmental process. Lastly, we have the site-specific integration system. And as the name implies, it's a single specific integration. Now, this can be done in two different ways. You can either utilize CRISPR-Cas9 and then have a sequence that it recognizes, and you integrate your gene into the genome. Or when it comes to cell line development, more commonly used, you will have a leap-in system. So you have a host cell that already have an integrated part within the genome that can receive the gene of interest. And this region is then usually selected for being a high production site. This will then generate an even tighter cell pool in terms of production, and you can then have a very low screening process when you are selecting your production cell line. Now, these different systems all have then pros and cons, and I will just briefly go through them. So this is usually, if you contact the cell line developer, this will be the thing that really deviates between them, and that is what type of system that they're using the system is quite often related also to the host cell, because the host cell is quite often then engineered. So when it comes to random integration, there's a quite old IP landscape, meaning that the license for these cells are usually much lower. You will have a much broader variation in your cell pool, but if you have a good screening system, that might not be a problem. Thousands of cells, then you have the capability of finding really high production counts. And I think different actors might say different things, but studies have shown that to find a really high production cell counts, random integration will still be your method of choice, because you can find, if you screen a lot of cells, the really top clones. However, usually these top clones are due to concatenames, meaning that you have multiple genes coming one after one another. And this also usually means that you have a lower stability, meaning that production will deteriorate over time. With this semi-targeted, the transposase version, you can have quite high titers more rapidly. You don't need to screen as many clones, and you generate a pool material which is much more reliable, meaning that you will the pool material will be very close to your single selected clone at the end. But here there's a lot more licenses and cost related to the system. And then lastly, with the site-specific system, you have a very high predictability. The pool that you can use will generate very similar material for the selected clone. So you could potentially do a lot more already on the pool stage. You almost don't need to do any screening. It's a very low amount of cells needed to be screened to find the perfect production host. But since it is a protein of interest is not expressing well at that site, and this also comes with quite high costs. Then for the second part that is important to think about is regarding the selection phase for your cell line development. And that is because monoclonality is patient safety. I'm not going to go through the historical reasons behind this, but the FDA and EMA follows the ICH guidelines that states that your master cell bank needs to be derived from a single cell. They actually don't say that it needs to be derived from a single cell, but they say that you need to have proof that your production host will generate the same product over an infinite amount of time. So then the golden rule is still that you need to have a proof that your production material generates from one single cell in the beginning. And this can be done in two different ways. Either you have image proof of monoclonality, or you can sequence the cell in order to prove that it comes from one single cell isolation. But a report on proof of monoclonality is needed. Okay, so that was just briefly background development is. I thought I'm going to give you what we at SciLifeLab now offers in our cell line development facility. So first and most important then is that we have now a license via Merck Life Science for their chosen platform. And that is then approved GMP cell lines for the GMP production of whatever recombinant protein. This is a platform that we have selected due to many different reasons, one of them being that it's a well-tested platform. They have been used in more than 80 projects, going through many different IND submissions. They're in phase 1, 2, 3, and full commercial, and very well known to the medical agencies. We also then have, of course, full traceability and cell line documentation on the two different cell lines that we have. So both of them are a K1 derived cell line, the chosen K1, and the chosen double-knocked GS cell line. And I should have mentioned in the transfection and selection process, you have then some type of selection pressure put on your host cell to integrate the gene of interest. And this can either be antibiotic or metabolic. And in this system, we use the metabolic selection system, meaning that for the chosen GS cell line, both of the GS or glutamine synthase genes have been knocked out. So we introduce our gene of interest together with the glutamine synthase protein, meaning that if we remove glutamine from the media, we will select out only cells that have integrated our gene of interest. Now, for the chosen GS cell, you usually get quite high production titers, and it's stable over 60 generations. For the K1 cell, you get a bit lower production titers, and here you still have the GS genes. So there you need to add some type of selection pressure in order to screen through your clones. But very important for these two cell lines is that they are both royalty-free. So it's very clear what you're paying when you're going into the commercial phase with these cells. Now, as I mentioned, the other really important part in cell line development is the proof of monoclonality. And we now have a very nice high throughput single cell cloning system with the upside from Cytina. So our process, we have the capability of screen multiple 384 or 96 well plates that will have a proof of monoclonality for 99.99% of all the cells that are printed out. So basically you have a plate that you put into your upside, you start it, and then it prints out one full 384 plate with one single cell in each well in less than an hour. So we can quite easily screen thousands of cells with this systems. It's also a very gentle cell dispension, so a lot of the cells will actually survive. If you have a fax system, then quite a lot of the cells will not survive. And if you're using manual pipetting of your cells, you will need to go through either images via a microscope or to do a dual rounds to get the full monoclonality. And I thought I'll go through a bit more on the monoclonality reports and how we can provide it with the upside, because it's such a crucial part in the cell line development. So with this upside cell dispenser, we have 3 ways of proving that you only have one single cell in each well that then later will become your production host cell line. First, you take an image here of the part that is being dropped down into the well before the cell is approaching, and also when the cell is within the part that is being dispensed, and also of course afterwards that the other cells are not being. Furthermore, when the cell is sort of falling down into the well media, we take Z-stacked images, and then we can see that it's only one cell falling down into the well. And then lastly, we take before and after images of the well bottom, seeing that firstly, there is no cell, and then there is a cell at the well bottom. So it's a very robust system of proving monoclonality in our cell line development process. Furthermore, we can then screen quite a lot of plates with the system, because it also automatically screens through the plates throughout the growth of the cells. So then we can select firstly on cells that are growing, but also importantly, another part that we have in our cell line development facility is that the Octet 96 system enables us to also measure titer at a quite early stage. So we don't only screen for cell proliferation, but we also early screen on productivity. And we do not need to screen productivity with tedious ELISAs. We can do it in a more automated fashion with the Octet 96. And I should just add here that for fun, I tried to do some AI-generated image to make our machines look more nicely. That is, of course, not how it will look when I measure antibody titers. Okay, so we're more going into our workflow. Is that we first start with a small in silico expression optimization. So looking at your sequence, if it's an antibody, looking at both the light chain and heavy chain to find the most optimal version of it. Then the genes will be bound into the FDO plasmid provided by Merck Millipore. And then we will go into mini-pool establishment before the single-cell cloning. Now this enables us to reduce the number of cells needed to be screened in order to find the really high-producing clones. After that, also a stability study in shake flasks will take place to make sure that the selected production clone continues to produce over a long period of time. And we can, of course, do all of the different analytical checks needed in the cell line development. And we could also provide a small-scale protein production batch for the people using our cell line development service. However, since we have such a high throughput cell dispenser, we can, of course, skip the mini-pool establishment step if timelines are very crucial. So it is possible to shorten the timelines here. And we also see that it can be good for some projects to maybe only use some parts in our cell line development services. So maybe they stop at the mini-pool establishment. We can keep the mini-pools in our freezers, and then they can continue once they are needing a single clone isolation. So I think we're quite flexible in what we can offer here. This is then a completely new facility. So it is, of course, important that we show that we, in the same way as for our drug discovery parts, that we can deliver on an industry standard for this. So that's why I'm very glad that we have this Bridge Sweden project. And we have 2 projects that already have received full funding to get the cell line development through our platform. And it's 2 very relevant antibodies that have also gone through the discovery phase within the DDD platform. So we are at the moment quite busy to develop and deliver well-established and documented clones to these 2 projects. And sort of to summarize what we have here then is that we have a new platform capability at SciLifeLab, where we say that we will provide the industry standard cell line development facility. I really want to emphasize that we have a quite high throughput, gentle cell screening capabilities. The upside can, of course, be utilized for other parts than cell line development as well. So it's something that I think we will use quite a lot. Most importantly is that we have 2 license for 2 different established GMP-approved show cell lines. I cannot go into pricing here, but I think we have a very good license offer. So the way that it works is cell line development activities have a certain cost, but then when you take over the cell line for your GMP manufacturing, then you, of course, have a license to the cell owner. And this is a license that quite often is related to different royalties. This is a royalty-free system with a very clear license structure that I think is beneficial for potential academic projects here in Sweden in the future. Within this Bridge project, of course, we have close collaborations with all of the different actors, but I want to highlight the collaboration with Testa Center. The 2 projects that I mentioned here will go through our cell line development and then go into the Testa Center facility in Uppsala, where we will do an upscale production of their host cell. And I think this is something that we can provide also future projects if needed. And this is something that we then have added to the drug discovery and development platform that can be used for our own drug discovery and development projects, but we are also a contractual facility for academic companies or academic projects. So please feel free to contact us if you want to know more regarding our cell line development facility, and also contact Alexandra Patriksson at Testa Center if you have questions regarding BRIDGE Sweden or the DDD platform. I think that was it. Was I too quick, maybe?
But then we'll have some time for some questions, so that's perfect.
Yeah,
Thank you very much, Niklas. Like I said earlier, but those who join in later on, we are recording this, so if you don't want to be included in the recording with your questions, please contact me via mail. So now I'll open up for questions. Just raise your hand and we'll hand it over to you and to Niklas to answer. Any questions?
And it can be any easy questions regarding cell line development. I think that's also the scope of this webinar, to sort of spread knowledge on how it works, etc.
Absolutely. Don't be shy. Jens, please unmute as well.
Yeah, hi, thank you. So great to talk. How long does it take from beginning to end?
So I think maybe I didn't go through it that thoroughly. It's easier if I share it again and show it. Yet again, that depends on what parts you want to include, right? But from...
Well, I guess from you take on a project until you deliver what you want to deliver.
In terms of the cell line development part, this is the timeline if we are at the moment that we can take them in. So roughly 12 to 20 weeks from start until delivering a RCB cell pool, I should say. So we don't do any master cell banking. That would need to be done at the GMP facility. But there's a lot of variation on how long you want your stability study, or if you want to skip the mini pool establishment. But I would say 16 to 20 weeks from start to delivered cell line.
Thank you.
Thanks. Paul?
Hello. Thank you, Niklas. Very interesting. No, I just had a question which is maybe more biological in nature, but have you any experience of like how well things like, post-translational modifications like glycosylation and things like this are kind of, recapitulated with these CHO models? I'm just thinking that if you're not specifically interested in a biological in terms of a therapeutic, it may be more as like producing biomaterials like extracellular matrix molecules. Is that something you have any experience of, looking at things like glycosylation and how well they're kind of captured?
Yeah, and that would be maybe more related to a DDD project then, but we do that as well, of course. So we have the license for two CHO cell lines here, which is for the full license for GMP capacity. If we're doing other types of material, which we also can do, then it's quite often under a research license. So then it's no limitations. So we routinely produce proteins with hex cells instead, because we can have a different PTM profile. But I would say we are very used to working with different difficult to express proteins, etc. And for here, that's also one of the reasons that we selected the random integration to hopefully have a bit more. If we would have projects that are maybe enzymes, etc., it would be nice to be able to screen through a large pool of cells to allow sort of biology itself find the one that are having all of the different parts needed to have the correct foldingness and post-translational modifications. But I think it's maybe more related to this will be mainly antibodies, bispecifics, etc., that I utilize that I see that we will use this cell line development facility for. But we also have quite good experience with working with other difficult to express proteins. So in our discovery pipeline, we always express all of our antigens ourselves, all of our phages and Fabs and full-length antibodies ourselves.
Gotcha. Thank you very much. Thanks.
I just received a question from the chat from San Lee. I'm wondering if we can use this platform to introduce copy number alterations.
You can of course screen with the cells that I presented on how the copy number looks in the cell that you have selected, but we will not maybe work anything with generating our own leaping systems or site-specific systems, if that maybe was the question. But you can always, of course, look at how the allele distribution and copy number looks in the cell that you have gotten. I don't know if that fully answers your question. I think maybe it would be if one would see that you can use the UpSite, the cell dispenser, for whatever project you have. That is always a possibility within SciLifeLab to contact us to see if you also are interested in our sort of machine park and want to use parts of that. Just contact us and we will see. But it's always a matter of how much downtime do we have on the machines.
Great, thank you. Any more questions? I got the thumbs up from Lee, so I think she was happy with that answer. Any more questions from you guys? Everything is crystal clear.
I think I really want to highlight, if there's something, if we mainly have an academic crowd here, is to really, if you're ever in the phase of you have a project that is going towards generating clinical material, or this is something that you need to think about to get a cell line, try to be quite early, contact many different CDMOs that have cell line development capabilities or cell line development providers, but really look at the small parts noted in the contract on what is the actual cost, what is the royalty cost for the cells. Maybe the price looks quite okay, but it's to be determined what the cost will be to release the cells from the cell line development. I think this is something that we have seen that a lot of academic have no real from, and this is probably the most important decision moving into building a company or generating material for your clinical studies. Whatever decision you make in the cell line development, it will be with you for the rest of the lifespan of your drug project. Always, that's one of the ideas with Bridge Sweden to generate more knowledge within all of us. And you can definitely contact us just to ask some questions. I've already had a few academic projects talking with me just to understand how does it work, what do I need to think of, etc. And I think one thing that is very good now with our cell line development capabilities is the cost structure that we have. The royalty-free cell lines and the cost related to it. And there's also so much more than just the license fee for your cells. You also need to think about, okay, what is the media cost for the media related to these cells? And that is something that we have also taken into consideration, that it's off-the-shelf media that is hopeful. So there's a lot of important decisions to take. And there's also maybe quite a lot of offers quite far away abroad that might not be so good in the long run. So really think about this if you're in the process of getting a production cell line.
Just a question from me. I mean, what do you see as the biggest mistake that people come to when they come to you? What might be the biggest mistake that they've done before reaching your facility?
Maybe that they've already have a cell that they have themselves developed that was under a research agreement, and they have no legal power to get that, to actually use it. And then everything is of course possible, but I think the companies that then own the cell line really see a a way of putting a quite high cost on that cell line if you've already done all the work. And maybe I think not realizing how long time it takes to develop a cell line. There are some providers out there that are really fast, and if you have the money, it can go very fast. But yeah, I would say that's the two maybe major things.
Yeah, and with this new the new cell line development, what else do you see? Like what type of solution can you provide? I mean, what will be like the main hurdle that you just feel that we will come by with this new facility?
I think one major benefit is for our own drug discovery and development platform, that we at a much earlier phase can test the manufacturability of our lead candidates. So I envision that we will, when we're down to a handful of lead candidates, we will make stable pools for all of these candidates. That enables us to maybe do tox material at a much earlier phase, and we will also see how well they produce in the system that they will actually use later on. So say that you have 2 lead candidates that are performing equally well, and one of them are producing very well and one is really difficult to produce. That can help you in your selection process of which one to choose. And I think also if we add on a bit higher production capabilities at our facilities, which we are sort of discussing, you can get material, even though it's from a randomly integrated pool. If it comes from a very narrow pool in the beginning, you can get material for tox studies already on that pool, which would be very beneficial. And that would also be beneficial for other projects contacting us. And I think additionally, we have the way our contract looks with Merck is that no sort of big cost comes until the cell is moved from our facility to another facility. So you could potentially pause that move for a while until you have the money needed.
Great, thank you. Any more questions before we round this up? No. And of course, you're always welcome to get in contact with Niklas if you have any detailed questions, especially when it comes to the pricing. You will just contact him and he can give you an offer for this. And are there any more general inquiries around the bridge project? Please contact me or Alexandra Patriksson, and we will see to sort it out. I will just finish off by mentioning again, we will have a seminar the 3rd of March where we'll discuss regulatory questions with RegSmart. So you can put it in your calendar already. I will send out an invitation. We will send out the video from this presentation as well, and other information. So thank you very much. I hope you find this useful and interesting webinar. Bye.